All right, so I started recording. All right, so this week, let's go all the way to the beginning. Now, we are moving on to Animalia. You remember that? Okay, what's this? Oh, we're still doing this thing. Is this recording? What is going on here? No, I don't want to record because we're doing it on our Blackboard, and I hope this doesn't. Okay, yeah, I guess. All right, so since we are going into the Animalia group kingdom now, you remember we have different kingdoms. Last week we finished a plantae kingdom and a fungi. Remember that? So you just got tested on units numbers 17 and 18. So this week we are going to do unit 19, which have a mixed bag of everything, because we are going to do lower invertebrates and some protostomes. All right. So now I'll pull this up here too, so that we can use, everything in conjunction here. So in this lab, I'd say, since I posted it up kind of late, you guys have to go through the PowerPoint when I post them up, listen to the recording, and use the PDF to study along the way. All right. So these are what you should know by the time we're finished with this lab. However, we do not have actual lab where you could do hands-on. So it's going to be very difficult for you guys to, to visualize and to feel and to know all these things. So here we have how many pointers or how many things you should know by the end of this lab, like 15 to know pointers. So here now, in this lab, we'll do the animals, and we're doing an introduction to lower invertebrates, and then we'll go on to protostomes, lineage. All right, so we'll cover all these things. Now, for the animals, we have these features or characteristics that we classified in the Animalia kingdom. So, of course, they're heterotrophic. They do not make their own food, like photoautotrophic, or, even chemoautotrophic. So here, this is a condensed version of those pointers there. So animals need to move around so that whether at one stage in their life they are mobile, but they're sessile as adults, but at one stage of its life, it has mobility. Now, they have to use their senses to perceive where they are in the environment and other objects. So, like when we see the flatworms later, they have certain senses that they will know whether to seek darkness or light, and, other objects where they don't bump into objects where they can get injured or whatever it is, move away from predators or chase after their prey. So they will need muscles to allow movement, and they have the nervous system in conjunction that they would use that allows them to move and incorporate what they have, the stimuli or whatever senses that they get to allow them to take action or no action. So they do have determinate growth as compared to plants. They had indeterminate growth. And what they have is that they would have tissues which are organized into organs. However, when we do the sponges, they are considered parazoas next to the animals. So they do not have organs, but they have tissues, a confederation of tissues or cells. And in the animals, they have a gametic life cycle where the gametes are the haploid part of its life cycle, and the adults are diploid. All right. So let's move on. Now, I think you guys have covered this in your lecture. So in early development, the types of germ layers, the number of germ layers will determine whether they are diploblastic or triploblastic. So starting from here, the zygote. So this is egg and sperm to make, a diploid organism where it will develop. It will go through early cleavage, where Going through the cleavage stage, it will go into blastula, and then it's a hollow ball of cells and gastrulation, and then we'll see the different parts of this kind of early development. So in the early development, in the diploblastic organism, they will only have ectoderm and endoderm. All right, so this is diploblastic ectoderm and endoderm. Wait, let me see if I could do, where was it? I was doing slideshow where, oops, sorry. I just wanted to see if I could do the pointer. Not record, but put this in that stage, but I'm not recording. Let's use this. Aha. Are you guys seeing the screen okay? I'm not recording this as per se, but I'm doing it in the Blackboard recording. I can't see your screen, so you have to tell me yes, shout out to me. So this will assist. I just recognized that I could do that or remember. Okay, so if the animal, this organism, only have two germ layers, which is ectoderm and endoderm. So you have the outer and the inner, which is endoderm, dermal cells, germ layers. So that will be diploblastic. However, the triploblastic animals will have all three germ layers or all three tissue layers, which consist of the ectoderm, the mesoderm, and the endoderm. The ectoderm gave rise to the nervous system and epidermis, and the endoderm, we are talking about diploblastic now, will give rise to the midgut and some inner organs of the vertebrates. And now if it's triploblastic, they'll have the additional germ layer, which is called the mesoderm, and that gave rise to the blood, muscle tissues, and which is like bones for vertebrates and some inner organs. All right, so let's move on to the next slide. So there are different kinds of symmetry of these animals. For instance, there is no symmetry at all, which is called asymmetrical. So it's like sponges, which we'll cover in the next, when we start doing the parazoas. We have radial symmetry, so you could cut it like a pie, like, in a pizza pie. So you get 8 slices of pizza, so all of them are the same. So it's radial symmetry. And then we have bilateral symmetry, where, it goes on to the medial sagittal dissection. And here, it will get a mirror image of the, left and right. So it will go from anterior to posterior and dorsal through ventral, all right? Now, I wanted to show you on my word. So when I'm doing this, so now, in these animals, this cavity in the animal is fluid filled. Either it has a coelom, which is called coelom. If it doesn't have a coelom, it's called acoelomate. Acoelomate means no coelom. They do not have a fluid-filled cavity. Our example here, which we are going to talk about today in the protostome, is the flatworm. All right, so acoelomate example here are the flatworms. Now you have the ones with a true coelom. So it's called a eucoelomate, or you could just call it plain old coelomate. All right. So the eucoelomate or coelomate consists of the annelids, mollusks, arthropods, echinoderms, and chordates. So we will cover these in the following weeks. Okay, and this is an annelid here, Glycera, and this is a eucoelomate or just plain old coelomate. Okay, so you see the coelom here, and why the pinkish area, which you see here and here. All right, over here you didn't see any. You just see that it has its 3 germ layers. So you have the 3 colors: the blue as the ectoderm, and then you have the mesoderm, this reddish color, and the green as the endoderm. See it here? So now in the eucoelomate, you would have the coelom, which is surrounded by mesoderm. All right, so this is a eucoelomate, a true coelomate. But if you have a coelom, but it's not surrounded totally by the mesoderm, this is called pseudocoelomate. That means it has a coelom, but the thing is, it's not like a purely surrounded by the mesoderm. So the pinkish part is the coelomic area, which is called the pseudocoelom, because you have the mesoderm on one side of it, and then on the inside you have the endoderm. It's not totally surrounded by just the mesoderm. So the example for a pseudocoelomate is the round worms. So these are nematodes. So nematodes are roundworms. Usually annelids, they call them earthworms. So the common names, we have to like kind of know the difference, whether it's a coelomate or pseudocoelomate or acoelomate. Flatworms, acoelomate. Coelomate, earthworm as an example. Pseudocoelomate, roundworms. All right. So let us move on to that was the type of coeloms. Now let's talk about the two lineages of triploblastic animals. So we have the deuterostomes. I'm pretty sure you guys covered this already. So the lineages is actually like the type of cleavage will have early cleavage, like cell division, and the fate of the blastopore, So in the deuterostomes, we have echinoderms and chordates. So in their early cleavage in the 8-cell stage, it is radial and indeterminate, which means when it's indeterminate, the early cells, each of the cell can develop into a smaller individual organism, all right? And then we'll look at the difference with the protostomes when we, once I finish with the deuterostomes. And the kind of coelomic formation is enterocoelous. It folds in from the coelom is formed from the pinching of the arch enteron. So you see the yellow part, and here pinches off, and then it makes the coelom. So here, another feature of classifying what lineage these triploblastic animals are going to be in are like, what is the fate of the blastopore? So in its development, the fate of the blastopore is anus first. So anus develops from the blastopore. So now let us look at a protostomic lineage of our triploblastic animals. Here in the protostomes, we have mollusks, annelids, and arthropods. These are our protostomes. So in early cleavage, in the 8-cell stage, the type of cleavage they would have is spiral and determinate. So spiral, they'll go, like a spiral staircase, or you see the DNA spiral like helixal. So it is spiral, and the cells are determinate. So the fate of the early cells are determined already. So if that cell was supposed to be an arm, it would be an arm. So if you separate it, it will not be another individual. And so it's determinate. Now, the kind of coelomic formation is schizocoelous, and it's the solid masses of the mesoderm will split to form the coelom. So you see here, from the mesoderm, the red, see how it's split? It's solid here and here, so it's schizocoelic. And then now, what is the fate of the blastopore for the protostomes? The mouth develops from the blastopore, all right? So in the protostome, mouth first from the blastopore. So just remember that, because in all the other exams, I don't know if you guys have gotten tested on this in your midterm, your first midterm, but if you didn't, this surely would be coming up in your second midterm, all right? So let's move on to our actual today's lesson for the parazoas. These are next to the animal, as in para means next to, zoa, animals. They do not have true tissues, but they have different kinds of cells. They have different kinds of cells, up to 34 different types, I believe. And when I go back to our PDF, our lab book PDF, we'll look at that and come back to our PowerPoint. And they have an indeterminate structure. So it's not like, like, let's say an animal, like, let's say, for instance, humans. Our determinate structure is, like 4 limbs, 2 arms and 2 legs. So it's a determinate structure, head, body, torso. And most of the cells are totipotent, which means they are the amoeba, but amoeba-site cells can become any kind of cell. So they can develop into oocyte, which means to make the eggs, or they could develop in the sclerocyte, which makes the spicules, and that's the structure that holds integrity of the sponge. All right. So here, these are example of sponges. Now let me minimize this so I can go into our PDF. All right. So wait, how do I do that? Escape out of here, if it allows me to do it. It's taken a long time. My computer is slow because I need to do some cleanup. And maybe I have to want to junk in it open up. All right, here we go. All right, so let me show you the screen here. So we covered this, the different types of characteristics and features of our animalia. So here are the different kinds of symmetry. So figure 19-1, which we talked about. Okay, I'm going through the pages, so at least some of you guys see this. And I'll just say this is bilateral symmetry, going down the sagittal, medial sagittal plane. So medial sagittal plane, and you get mirror image. And then it tells you the different positions. In the belly side of this frog is the ventral side, so you need to know the orientation of your organism. The frontal part is called the anterior. The back part is called a posterior. And the lateral and medial is in the middle, when you cut down the middle, so medial. Dorsal is on top, and the ventral is the belly side. Yeah, proximal and distal. So when you're doing other identification or positioning your organism when you're doing dissection, it's very important to know the animal's orientation, because you will need to know that for your clam dissection and your oral quiz and your final practical. All right. So we talk about the kind of coeloms, the body cavities, the coeloms here. All right, this is a recap of what I just showed you guys on our PowerPoint and the different types of animals here. We'll talk about this. So now, let me show you the word I have here because it's less fussy. I was practicing my, print screen and also my pasting and all that stuff. Okay, let me make this slightly bigger. Okay, so this is the phylogeny of the animals that we started to talk about what classified as animals, the characteristics, and of course the kind of coelom it has, and to determine whether it's diploblastic or triploblastic animal, and whether it's in the diploblastic, in the diploblastic animals. We'll cover this today, which means 2 germ layers, the ectoderm and let me say that again. In the diploblastic animals, the ectoderm and endoderm germ layers. In the triploblastic, which are bilaterarians, which means they have bilateral symmetry, they have all 3 germ layers, the ectoderm, mesoderm, and the endoderm. And in the trophoblastic lineage, it's further divided into the type of animals that they can be protostomes or deuterostomes, dependent on early cleavage, and then also the fate of the blastopore. All right. So our common ancestor here from the animal, for the animals in our phylogeny here, we have this other group, which are called the sponges. These are the parazoas, which means next to the animals, and they're called porifera also, which means poor animals. All right, so all of these we'll cover for the rest of the semester. Today we'll cover the sponges, we'll cover the diploblastic animals, and some protostomes in this clade here. All right, so let us go back to quickly look at this. All right, so I'm trying to correlate. And here there's something, a small write-up, 19-4, to refer to this here, that ctenophores are diploblastic. But then again, they were saying that they could be triploblastic because they have muscles. So this is a write-up here in edition, the second edition of our lab book, and third. Since I have the first edition, this was not added, that was not published in there. So this is another phylogenetic, how to call it, view of our animals and our co-relationship. Because you remember when we did the protists, choanoflagellates is a protist. So from here, this point here, the choanoflagellates, it's like not in the animal clade, in this phylogenetic tree. And here we have ctenophores, which is the comb jellies, and then the sponges, which we'll talk about, and the placozoas, and the remaining animals, which are the triploblastic. All right, so this is an addition to the book which I had in our first edition. So now we'll talk about the sponges. The sponges, they are called the poor animals. Let me see if I could highlight this. Highlight. What is going on? Come on. What is going on? All right, let me type it in so that we could do the. So in sponges, so I have all these things. You guys got to take notes, all right? Because this is very important, that this is how you will study well. I want to start a new. Okay, so now we already talked about the lineages, the kind of, let's say, we talk about coelom, right? The types of coelom, types of, oops, here, even type now. Okay, what's going on? Of them, which is, so this is how I want you guys to interact and take notes, because it's kind of hard teaching you guys stuff and bringing everything together. So we have acoelomate, or type of coelom, acoelom, or the types of coelom or coelomates. All right, coelomate means that, the coeloms that they possess. We have pseudo. Did I spell it right? I never spelled this word. Coelomates, right? Or they have pseudocoeloms. And we have the eucoelomate, or you call them coelomate. Simply, you could take off the EU from here as, means true. So this is how I want you guys to take your notes, all right? So these are the types of coelomates that we have. And then we said the types of animals we have according to their germ layers, right? So we have diploblastic, which is consistent of 2 germ layers. I'm not going to go through all this again, right? Triploplastic, which has all 3 germ layers, right? And here within the triploplastic, we have the lineages of either protostome. It consists of protostomes and protostomes. All right. So as we said, it depends on its early cleavage and the fate of the blastopore. So now we're coming to the animalia. So this is how we're doing the animalia. So we are going to do the sponges, as we're saying that the sponges, these are the terminology. You need to know these terminology because 99.9% your final practical would have questions pertaining to this. All right. So they're called core animals. They're also called parazoas, which means next to the animals. And they're also called, as I say, animals was porifera, all right, or poriferans, if you want to call it. All right, so these are the 4 animals. That's how they came about, these words. They're not truly animals if you're going to see it like that. So the types of sponges we have now, according to its spicules. So you have to go through step by step to do all this stuff. So these are the parazoas. There are about 10,000 species of these things living in salt water, which is marine habitat, or fresh water. So what gives it its structure when we see the sponges after they die are the spicules that's remaining. All right, we'll talk about the sponge, how they are. Where are the sponges? Oh, sorry. I went too far here. I need to come back here. So here now, the types of sponges we have are glass sponges, sponges, and calcareous sponges. All right, so this is where, see, I'm repeating all the stuff here that I would talk about, but you need to know really seriously. So the type of sponges, according to its spicules. All right, so we have calcareous, most sponges, and glass sponge. Let me see if I have it right, because like I may call them different names. It's kind of hard remembering this stuff, especially when I have to go between books. Okay, so this glass sponges, most sponges, calcareous sponges. I just flip it around, so it's still the same. All right, put it in so that you guys just don't mess it up. Okay, so what we have here, glass sponges, it's made of silica, right? You remember, silicaceous kind of spicules? So spicules, we'll talk about those. So that is silica. This is proteinaceous, different type. We'll talk about those too. Proteinaceous. That's spell a word. Well, it's kind of close. I'll have to go back and look at it. And organic spicules. We'll correct that when I go back into the PDF, because it's like going back and forth. It's kind of like hard to remember. So this is made of calcium carbonate. Remember when we had the foraminiferans, calcium carbonate? So this calcium carbonate, this kind of calcareous sponges will be in shallow water, and that's marine. And this here could be freshwater, marine and freshwater, where they could be found. And this one is in deeper depths of the ocean water. So the reason the calcareous sponges cannot exist in deep waters is because that they will dissolve. Same thing like when we talk about the other groups of protista, you remember we talk about forams. Forams are made of calcium carbonate, and they are the deposits which we see in the white cliffs of Dover. However, when we talk about diatoms, they are made of silica, silica, siliceous material, so they could be in deeper waters. So diatoms and glass sponges can be found in the deeper oceans. Demo sponges are found in marine, both marine and freshwater. Their spicules are a combination of proteinaceous and organic matter. So let us look for this here. When you're reading your information, we'll see the different kinds of sponges that we cover. Glass sponges here. Euplecta is our example. So when you go to your pages, you read through these things. Spongenin fibers. This is the one I was going to do. So they could have protein or siliceous spicules. That's what I needed to put in here. I spelled this wrong. Spicules and spon. Did I spell it right? I might have it wrong. It's like I couldn't remember how to spell it. Spongin. Okay. And let's see, protein. So that's protein. All right. So this allows it to have different kinds of stuff. So this is like protein-like. So this is a combination of this. So what are spicules? Spicules are this kind of structure that gives the sponge its shape and integrity. It supports the architecture of the sponge. All right. So when you get a sponge that's, a bath sponge, you would see that it has a structure, but the animal has already died. The cells. So in this here now, let's go back to our PowerPoint. So you see this, the different types of cells, but the spicules, which is the structure that allows it to hold the integrity of the sponge. So now here I have the types of spicules that we would have looked at in our lab if we were to do real lab. All right, so these are pictures I took in class, but it's sad I didn't get enough of them. So these are the slides, which would have been in the slide boxes. So here will be the Grantia spicules, whole mount. And then we'll have the Grantia, which is that sponge, and it's also called cipher. We have cross-section and longitudinal section. So let us look at those pictures I took in class. So here, okay, let's make it a little smaller. So this is what the spicules will look like if you were to get a slide and look at it. These are spicules of the Grantia. All right, so these are Grantia spicules, and since this is a name of the genus, or it's also called cipher. So it is what do you call it? That's the other name for it. Species. All right. So this is what you would see in lab if you were to use a slide and to look for it. If you see this, pay attention, class. If you see this in your final practical, you better know what it is because it's very important. So now, this is in what phylum? It's in a Parazoa, Poriferen phylum. So you got to make sure you know all the stuff that pertains to this spicules of the sponge. All right. So now let us go back here. Well, hold on. When we do the sponges, I'll show you a cross section. This is a cross section of the sponge. So this is cross section, and below here, this is from the lab picture that I took. This is longitudinal section, so you're cutting it lengthwise. All right. So let's go back to our slide here. All right, we don't need this. Now, this is talking about all the attributes that they have. So sponges are a confederation of cells. They're not multicellular, all right? And it's not colonial in such a way. So this is a basic architecture of the sponge, where it looks like a vase. The simplest one, we'll see the different types of complexity of the architecture of the sponges. But this is the simplest, form unit, which is vase-like. Now, what happens? Why do they call it pore animals? They have these special pores that's called porocyte. This word here, porocyte. Let me see if I could do the slideshow here again, which current slide. I'm not recording, I'm pretending. What is this pen? Okay. So the porocyte here, you need to know that. That's why it makes it a pore animal, right? Porocyte. So these are special cells that water goes through the porocyte. It's like a pore. This is the water going in. Once it goes into the sponge, just this empty space, that's just called atrium or spongocoel. All right, so this here, atrium, spongocoel, you need to know that. So here now, in the inside of the sponge, the interior, you have special cells that's called choanocyte. All right, so in a porocyte, water comes through it, and then it brings in food, all these particles through in the water. So the choanocyte, it is named so because of it looks like the protist choanoflagellate. So here the choanocyte, we'll see what it looks like in a bigger, more detail. These special cells are lined on the inside of the sponge, and they will use a flagellum and bring the food into this collar of cilia, and they will digest the food. And once the food is digested, it will, well, not digest, let's say breaks down the food, ingest and digest, and the food will be taken throughout the mesohele. So this is diploblastic now, mesohele. This is not mesoderm. Mesohele is a gel-like matrix that allow the other cells, such as amoebocyte, which is totipotent, to move around throughout the structure of the sponge. Now, what is the exterior of this sponge? They have a special lining of cells, which they're flattened, and they're called pinacoderm or pinacocytes. So here you see it's flattened, and the special cells, which are the porocytes, embedded, throughout the exterior. And these are the different kinds of cells or structures that we are looking at. Now, once the how does it hold a structure? The spicule. So the picture I show you, spicules will maintain the integrity of the sponge, and it's good for protection too. So if a fish want to chomp down on the sponge or graze on it, the spicule, like whether it's made of calcium carbonate or glass or proteinaceous spongin, this will deter the animal from, grazing on it. So this is the spicule that you would see here, and I showed you the picture before. And we talk about the atrium spongocoel. So this is the so-called empty space where water goes in. And here now, so what happens to the water? It can't keep taking in water. It has to have an out current. This is in current pore, water comes in. So after it gets its food and all that stuff, flushes out all the other, wastes, it will go through something out the open, which is called an osculum. All right, so osculum is the opening that faces upward. And there's a lot of other spicules around here, around the opening. So this is a cutaway of the sponge that we are seeing in here. All right, so you need to know these words. Osculum is where the water comes out. The water comes in through the porocytes. So that's the incurrent pore. Pinacoderm is the outer, the cells that on the outside of the sponge, and inside it's lined by choanocytes. All right, there is no mesoderm. There's no mesoderm. There's a mesohele, which is a gel-like matrix that allows other cells, such as amoebocyte, to travel to take food to other cells. And also, like amoebocytes are totipotent, so they can be any kind of cell. They can become any other cells. They can become sclerocyte, which makes spicules. All right. So I have covered this. When you guys get your question in your final practical, you better get it correct, because there will be 3 questions out of your 60 questions. And, it's like 1.67, I believe, on that. So it's like times 3. So 3, that's like about 5 points. So you don't want to mess this up. All right, so let us look at more details, which I've already spoken about. Again, this one, if it says silica spicules, so it's most likely glass sponge or demosponge. Amoebocytes are the special cells that it could become. I shouldn't say special. They're totipotent cells, which can become sclerocytes or whatever. So here we say many pores. So this is only taken in. It doesn't expel water. This is wrong here. All right, it only takes in. All right, I should correct that. All right, so here we say pinacocytes, again, like skin. The pores. The other cells are porocytes. Choanocytes are the color cells. So this is the choanocyte here that lines the inside. And this is, it looks like a choanoflagellate, but it is not. Choanoflagellates are protists. Here, choanocyte is the cell, the feeding cell inside of a sponge. So this is choanocyte. So you see the flagellum, and you see the color of around the flagellum. All right, and here the amoebocyte, they move around in this mesohele. All right, on the inside. And what else we have here? Okay, so we have ostia, ostium. I think ostium should be singular. Ostia is plural. So ostium or ostia, if you see either of these words, this increases the surface area of absorption. All right, so let us see what we're doing here. Let's escape out of here. Oh, we were recording. I didn't realize that. Okay, let's come out of this one so we can look at more of the other words, like the structure of the sponges. Oh, they don't have that much there. I skipped ahead. So here are examples what spicules look like. And the type of spicules will determine the type of sponges they are. These are the different kinds of cells. So there are in the spicules for the cypher or the grantia. Grantia and cypher are the interchangeable name for those sponges. They have T-shaped, short and straight branch spicules or matrix of mesohele or mesenchyme. All right, so these are interchangeable. As we say, excurrent comes out, which means the water that get expelled out is through the osculum. amoeboid cells, which are called archaeocytes or amoebocyte. So this is interchangeable with amoebocytes. All right, so here now in the in current, let's see, to increase, this is a bigger version, more detailed version, pinacocyte again. An oocyte is this special cell that makes the egg. And this here, spicule is made out of sclerocyte. So this, you don't need to know so much of detail into this. Archaeocyte is also called amoebocyte. And the in-between matrix that they float around, they swim around in or move around is a mesohele or the mesenchyme. And what else we have? And the special choanocyte feeding cells. So this is more details at the different types of cells. I believe there are up to 34. Now, when we said the pore that the water comes through, they are called porocyte, right? So this here is a porocyte. Here is a porocyte. But this word ostium, why we need to know ostium in conjunction with porocyte, to increase the surface area of where the water can come in. It has this kind of, let's say like this, so it increases the surface area, right? So when you say you have these kind of grooves like ostia, it increases the surface area for more water to be brought into the inside of the sponge with the food, which the inside of the sponge is called spongicle or atrium. All right, so this is the sponge in its functions and its cells. Now, the sexual reproduction here is you have the egg and the sperm. So the sperm from another sponge will be released and it will be taken in through the porocyte, right? So the porocyte, let's say sperm comes in here. And then what happens is this special cell, which is called an oocyte, is as an ovum. It will fertilize it. And once it fertilizes it, it will make the larva, the embryo or the zygote, and it will make the amphiblastula. This is a special name for the larval stage, which is dispersive. So you need to know that in the sexual reproduction, it has the sperm and the egg, the sperm from another sponge, which is released. It is taken in through the porocyte, and it will fertilize the egg in the ovum here in the oocyte, and then it will develop into an amphiblastula larva. This is the only dispersive stage of its life. So you remember the animals has to be able to move. So this is the dispersive stage. All right. And the thing is, you need to do the read up on it that, let's see, dispersive stage. So these are different forms because adult sponges do not move. They are secured at the basal end onto some kind of substrate. So what is the dispersive stage of the sponge? It's only when it is in the larval stage, and that is called this thing here. You need to answer that. So here is where your answer is. Amphiblastula. So if you guys don't answer that, I'll be very disappointed in you guys not doing your due diligence on reading. So the larval form is the only time that it is dispersive. So you need to read all the stuff. It's a lot of work, but this allows you to understand the work better. All right. So let's move on to the other slide. It's 3:13 already. Okay. So we covered all these things on the sponges. All right. We talk about the different kinds of spicules. So this is another slide of we got on the internet. Somebody has a question. Let me come over there. You're not seeing the slides, Edina? Oh no, what happened? Did my business die off? Refresh. Okay. Abdullah, you refresh. Is it okay? Oh man. Okay, let me know if you guys did. Sometimes, you know, when Blackboard, when everybody's on, like the whole of Queens College, it didn't work. I wonder if you could come out and back in. Do that, each of you guys. It might have knocked you guys out. Okay, yeah, Fahib came out. Let's see if he came back in. Let me make sure I'm recording. Let me see. Is it being recorded? I'll be so mad if the recording got knocked out. Okay, yes, it is. Sorry. I just wanted to make sure that, okay. Yes, thanks, Adina. Okay, are you guys seeing it? Can't see anything? Screen is gone. What's going on? Okay, time to go home. Oh, you guys are home already, you know. Okay. You know, I should have done the recording and you guys look at it on your own time. You guys don't see anything at all? Heck, maybe I have to refresh mine. Wait, hold on. Let me go and see if I do share screen, because I know I didn't touch anything unless my screen decide to do some crazy thing. Share application screen. Okay, let me do it again. Anything? Hold on. How about now? Okay, great. So what I did was came out and do back the share again. All right. So I was just yammering away and it's like, hey, nothing is going. So did you guys see the screen or it just happened? All right. So now this is more details which you'll see. So the amoebocytes are the ones, it should be AMOE. You know, that's spelled a little wrong. They make the skeleton as either calcium carbonate or silicon dioxide, and as a spongenin, which is proteinaceous kind of compound with silicone. That's silicone. Yeah, silica. So I think this should be silica. Okay. And these are the spicules. These are the structural support for the sponge. So some of them are separated and some of them are stuck together, but it will reinforce its integrity. All right. So now we were talking about vase shape of these sponges. You know, the basic is a vase-like shape. You see the arrow going out, that's where the water goes out through the osculum. All right, osculum here. So this is the asconoid. You remember we did the ascomycota fungus. So asconoid as in a cup shape or vase-like shape, like this, ascus. So they call it asconoid. So this is the simplest architecture of the sponges. So asconoid is just like this here. Syconoid is the shape where it's made groupings of these structures. So you see it gets a little more complex. So this is syconoid. And now the most complex of these groupings are made up of the leuconoid, which is made up of syconoids structure, syconoid-like structures, all right? So you see how you get the simplest asconoid. Then you have the next complex, which is syconoid, and then the most complex is the leuconoid, all right? So this is the basic structure, but they have indeterminate form, but this is a basic. So we said that it will continue growing, so it doesn't have any kind of symmetry. So if you ask the question, what kind of symmetry it has, either none or asymmetrical, all right? Okay, so now enough about sponges. So after that, we should go get some sponges and go down to the beach in Miami Beach with all those party goers. But that was kind of not so responsible. So now let's move on to cnidarians. This now, cnidarians, we are moving on to the animalia that's diploblastic. Sponges are not considered diploblastic because it's not really any kind of layer, really. You see the layers, but it's a confederation of cells, all right? So now let's go to this group here. Coming back up again, if I could find it. I wish I could have the screen that switch me back and forth because we have so much. Okay, so we covered the sponges, the 3 different types. Now we're going to do the diploblastic animals, which is 2 germ layers, ectoderm, endoderm. However, there's a notation now that they added ctenophores might be considered triploblastic because they have true muscles. But now we are going to do the group of cnidarians. All right, so let's go on to this. So cnidarians, if you are to go into your PDF here, let's go to that. So let's see what stops off at. Okay, so that's punching in. Okay, so the cnidarians, they consist of the hydras, jellyfishes, anemones, and corals. All right, and we have the class names here, which we'll talk about in more details as we do each one of these. Okay, so what makes the cnidarians, what makes them cnidarians is because they have these cnidocysts. They are diploblastic because they only have 2 germ layers, and they only have ectoderm and endoderm. They have a 1-hole gastric cavity. So you see it's like in and out of that same opening. So this will be called the mouth or hypostome. And here you see the mouth opening here. This is the medusa stage. So it has this kind of bell-like thing. The one to the right is polyp. All right. So here now what we have is a 1-hole gastric cavity. So food comes in, processed, and the waste goes back out. Same opening. All right. So it's a 1-hole gastric cavity. So it's 2-way, in and out, 2 direction. They do have how do they move around is that they don't have true muscles, but they have longitudinal and circular contractile fibers or fibrils. So they contract, the squeeze, and the opposing the longitudinal and circular fibers will allow them to move. Okay, what makes them cnidarians is because they have these special cells that's called cnidocytes. All right, we don't pronounce the C, so you say N, cnidocytes. So these are the special cells that we will see as the commonality of the cnidarians of 4 classes. What kind of symmetry they have? Radially symmetrical. So as say if you cut it like a pie, it will be like, if you cut this, you'll have radial symmetry. If you cut this, which is like bell shape or umbrella shape, you will have radial symmetry. All right, so let's go into the types of forms they can have and which is dominant, or if there is absence of either alternation of their generation, whether it's polyp, which is this. Polyp looks like, how should I describe it? Yeah, like a cup facing upward. And the open area where the food comes in is the oral end, so it faces upward. And the aboral end is the part that's secured to the substrate. And here now is the medusa form. The mouth is facing downward in this case, but we'll see in some of the examples where when they move around, the mouth is facing up, and they're motile, they can move around. And the medusa aboral end is on the opposite side, so aboral, the opposite side. Okay, this is a cross section to show you the germ layers. They only have ectoderm and endoderm, so they are diploblastic. They have this kind of gel-like matrix, which is called mesoglia, but it is not mesoderm. All right, mesoglia is not mesoderm. It's not a germ layer. This is a gel matrix that allows it to communicate and also to help it to move when it moves, squeezes the bells, these areas, so it allows it to movement for movement. Okay, so what makes this an Adarian? They have this special cell that's called a nematocyst. And this is a schematic of the three different stages of the nematocyst. When it is not discharged, it's in its resting stage. So what happened is, meaning it's not discharged, they have this special trigger here that's called nidocyle. And this, once it gets irritated or chemically or maybe mechanically or whatever, this covering, which is hinge, it's called operculum. It will flip open, and then this barb will shoot out, which is connected to a thread. And whether it is trying to capture prey or to defend itself, it can be very painful. So here, once it's discharged, it cannot be used again. So that's it. So it has a certain distance that it can, get to its prey or fend itself from the predator or any kinds of disturbances. All right, so this makes it a Cnidarian because of the special cell that's called nematocyst, and that's to the outside of the organism. All right, so this is the epidermal layer when you see the thickened area. So it's embedded in its matrix in here. All right, so let us go on to the next screen. So now we have four classes of our cnidarians. So we have the hydrozoans. All right, so we'll talk about that. We have the cyphozoans, we have the anthozoans, and we have the cubozoans. So the hydrozoans, you need to know this. This table, you need to know the characteristics, the life cycle, and the species that represent each of these classes, because it helps you organize your organism and study. So the hydrozoans will consist of like hydra and all of that. So it's like a hydra-like cnidarian. And they're found in fresh water and also in the marine environment. However, the freshwater hydra, which is like the brown hydra, they're solitary. They do not live in colonies, so they are solitary. So know this one. All right. And the thing is, they have feeding and reproductive polyps. So their stages are, so they have different kinds of polyps, the characteristics, but a predominant stage is a polyp stage. So in the medusal stage, it's very, very a short period of its life cycle, and that's where it's a sexual stage, all right? And some of them do not have a medusal stage. Our example here are the obelia, which we'll talk about. The hydra, we'll see Physalia physalis, that's a Portuguese man of war. So let us look at the hydrozoans. So here, the hydrozoans. So as we're saying, there's distinct alternation of generations where it is polyp and medusa. Many are colonial, but as I was saying, the brown hydra, which is freshwater, they're solitary. So this one, they're solitary. So they do not exist in colonies. Now, if we were in class, we would have, take a sample, a wet mount, and do it. And also we have, and then also we want to see if the nematocyst could be discharged when we get a sample and we put a little bit of vinegar in it, acidic kind of environment, and you have to look at it on the high magnification to see that the nematocysts are discharged. However, since we do not have real lab, I'll have to do a lot of talking. So let me show you a brown hydra in class that I have in the picture. So this is from the picture I took. They're pretty big, so it fills up like the whole screen, and this is at 4x. So if you made it like much larger, you will see it like more details. So this is a brown hydra this here is the mouth over here. So the way I took the picture, you know, the basal disc is on the other end. So this is the oral end, and these are the tentacles. Okay, so that's a brown hydra. It's solitary. And what else I have to add to that? We're coming back to here. Okay, so in the hydra, they can be hermaphroditic, which means they have both male and female sexual organs, gonads, all right? So here we have like a cutaway of the brown hydra, or of a hydra. Hypostome, this is like the opening here. Tentacles around it. And this is the body of the hydra. Peduncle is where it kind of stands on and it flips when it moves. So what it does is it could do somersaults to move about. So they are motile. They're not fixed in an area, as we'll see later on in the corals. And of course, the sponges, they don't move. They're more secure onto the substrate. And in the sponges, the only time that they are dispersive is when they're in the larval stage. However, here in the hydra, they can move about. So they can flip and move around, somersault. So now this is a cutaway of the hydra body to look at the different kinds of cells. So here, this is ectoderm, which means it's on the outside. This is in the outside environment. The special cell again, as we're saying, I'm not doing recording, but I just want to use this as draw. So here again, hematocyte, which you have to know, that makes it a cnidarian. Ectodermal epithelial cell, this is on the outside, which is ectoderm, and endoderm. So this makes it diploblastic. So the nematocyst defines it as cnidarians because all cnidarians have these special cells embedded in its ectodermal epithelial cells. And there are various other cells here, but we won't go into that. In between the ectodermal cell layer and the endodermal cell layer, we have the mesoglia. This is not the mesoderm, all right? Mesoglia is a gel-like matrix where, you know, like other kinds of cells could travel about, like to bring the food to other parts of the organism. So let us look to the inside of this hydra. So inside we have these special cells that line on the inside. You have gland cells, and you have all these neurons and all this stuff along the way here between the layers, and it communicates, you know, like to know whether to move away or whether it's feeding and all that stuff. So these are the endodermal epithelial cells. Those are on the inside. So now you have, this is a cutaway of our hydra, and it's a hydrozoan. So here you see the nerve system is net-like, all right? So it has a net-like nervous system, and here they have a concentration of the nerve cell around the mouth. So it's concentrated around the mouth, all right? So now let us go on to the next slide. So another example of hydrozoan is the obelia. This is colonial, all right? Oh, wait, I got to show you also. Hold on, let me show you that in our PDF. All right, in our PDF here, because I want to show you on the hydra that it's like totally hermaphroditic, or it could be dioecious too. So I just want to show you more details here. So this is, as it discharges of our nematocyst, the stinging cell. So it's also called stinging cell, cnidocyte. All right, so this you could look at more detail. And this is, I'd say, polyp stage. This is a polyp, and this is a medusa. They have this backwards. All right, this is medusa. Oops. I hell. This. It's medusa form is this one here. All right. Not this, it's flipped around, and polyp is this one. All right. So now put highlight. Okay, so I wanted to show you this too. Where is it, Hydra? Okay, so I just wanted to show you guys that it has here both, sexes. So it is hermaphroditic, or it could be separate dioecious too. But see the ovaries, right? So that makes it having both sexual organs for male and female gonads. So it makes a sperm, it disperses. I think it normally, it will not self-fertilize, but it may fertilize another hydra. So here, let's pretend it gets some sperm, like from another one, that it disperses in water. And then here, once the eggs are fertilized, of course, like they'll develop into a young polyp. And this sexual reproduction. When it buds, this is asexual reproduction. So budding is an asexual reproduction way of reproducing. So let's see if it says it here, tubular bud. So after fertilization, it gives you more detail here that you can read. A resistant zygote is capable of surviving harsh periods of development. And as we say, hermaphroditic, maybe, or it may not be. They could have separate stuff. And we said it's diploblastic. So these are the key words you need to know. Radial symmetry. And I'd say sexual reproduction is when sperm and egg, and then budding is asexual reproduction. All right. What else I need to tell you guys here? And then again, I say it's a one whole gastrovascular system in and out of that same opening in the mouth. All right. And there's a lot of I say the nerve network around the mouth area, so it's concentrated there, although it has a nerve network throughout its body. So you guys can read all these things, and it allows you to know more details. So this is more another cutaway version of the body wall of the hydra. Some more details like than the previous. And then you need to read all that. There's different kinds of cells. It's a little more detailed, but it's good to know it, because you never know what kind of questions you guys are going to get in your second midterm. All right. So this too allows you to study for your lecture exam. Okay, so now let us talk about the obelia as our other example. This is colonial. They have different kinds of polyps. You have the feeding polyps, which is called the gastrozoid. So here, let's go back into this thingy so I could kind of like draw onto it. So you have the gastrozoid here. I went too far. So here, gastrozoid. This is a feeding polyp. You see, it has all these tentacles that stick out here. So this is a colony where you have different types of polyps. So here, once it's feeding, it grabs the food in, brings it into mouth. It makes the food, and it just taken all over in this throughout the colony of polyps. And how it can take the food throughout is like, if this feeds, everybody gets fed. So it's a cenosarc. Cenosarc is, like this kind of tube that allows like a highway for all the food to go to all the polyps throughout the colony. So gastrozoid is the feeding polyp. And here now, the sexual reproductive polyp is called gonozoid. All right. So here you would see medusa or medusae that bud out of here. So what happens is this is sexual reproduction where the medusae are reproduced through here, and we'll see another screen. the reproductive cycle of the obelia. And here you could see in the medusal stage, they have this, it looks like an umbrella or kind of cup-like structure. All right, so now this is the schematic of the obelia. So here you have, it's in a colony, it has a stolon that keeps it anchored to the substrate. And here you have the different types of polyps in this colony. You have the feeding polyp, so you see all of the structure here that it brings in the food, sweeps in the food from the surrounding waters into its mouth. And here you could see this is a reproductive polyp, which is called gonozoid. They didn't put the name here. And this is another reproductive polyp, which is more detailed. So here it makes medusae as a plural, but medusa is a singular that buds out. And what happens is it will disperse in the water. And this is in the medusal stage where you could see the gonads in this sac here near the radial canals, where it's either male or female medusa. All right. The female medusa will make eggs. And what happens is the sperm, if it's a male medusa, it will make the sperm. And once they're fertilized, it will make, this part isn't right, it will become a planular larva. So this is important. This is the larval stage. This is the dispersive stage 2, where it goes from, medusal stage and the egg, and it will find a place which is conducive for it to develop. And once it finds a place, it will develop into another young hydroid colony. So let us come back here into our, if I was in class, into our thing here. So this is a more detail of, it's, I guess, more detail in addition to what I just showed you in the other PowerPoint. So I say reproduction of the polyp is asexual by budding. So you have to know that. And of course, the sexual here, gonozoid, that's a sexual reproductive polyp, right? You have to know that. The gastrozoid, when they have lots of tentacles, they are the feeding polyps. So these are the things you need to know. And the tiny buds of medusa, this is dispersive. So here you see the gonads here within, near the canal here, and menumbrium along here. Statocyst, it lets it know the orientation, and ring canal. So here you can see different types of medusae. You have the female one that makes the eggs, produce the eggs. You have the male medusa, which makes the sperm. And of course, here, sperm and egg will fertilize into forming the zygote. It develops into the embryo. And then this is what you need to know, the special terminology. It has cilia in this planula, which is a special name for the larva that's the dispersal stage of this hydrozoan. All right? So these are the terminologies you guys need to know. All those words I underlined there. All right? So when coming back to the feeding polyp, once it's retracted, it looks like this. If it's feeding, the tentacles will be out and about, trying to sweep in food into its mouth. All right? So now, let us move on to the next slide. Okay. So the next other example is Gonionimus. This here is a medusal stage. It has, I don't believe it has a large, it's a very small polyp stage or none at all. So let us look at the thing here. It does not say, but certainly do your reading here, and it gives you more of the Gonionimus. Obelia. Okay, go. One thing about the Gonianomus, they swim with their mouths facing upward. The others, they swim, with their mouths facing downward. So this medusa of the Gonianomus, so this is what we're doing now. You need to say that the bell opening, this is what surrounds the opening of the umbrella shape. Here, we need to know this word. And they have the pads, and then let's see, swimming. What other words we need to know here? And the band of tissue, which is called a vellum. You need to know this word, the vellum, which is a characteristic of the hydrozoans. So the hydrozoans have a vellum. The other cnidarians don't have it. Cnidarians have cnidocysts or nematocysts, but hydrozoan class of the cnidarians is defined with a vellum. All right. This Gonianomus, as I said, it swims facing upwards. Tentacle is facing upward. Okay. What else I need to tell you guys? So it catches all these floating vegetation and all that stuff. So all of these things, I want you guys to go into your PDF, do the reading, do your underlining and highlighting. I don't know if you print out your PDF, but I advise you to do so. It's kind of bad because, ink costs money. But if you were to buy the book, it would have been $140. So I don't know, like I say, which is better. All right, so this is our example. This is the cross cutaway section of Gonianomus, with it facing down. Suctorial pad, which is adhesive pad that sticks on to vegetation, and all that stuff while it is traveling about. And these are the other stuff. And these are where the gonads are found along the radial canals. All right, so you should have 4 of them. And you have the gastrovascular cavity and the mesoglea. Oops. Sorry. What else I need to tell you guys? Okay, so that's enough on the Gonianomus. Let's move on to the Phasalia phasalis. This is also a hydrozoan. These are the Portuguese man-of-wars. In Florida, you'll find them, and I believe here up in the Northeast, because if you see this along the shoreline, I'll sometimes have them with a stick. So this is a colony. They have different types. This colony of polyps, there are different types of polyps here. So here you have the stinging polyps, which means it's for defense or to capture prey. You have the feeding polyps, fishing tentacle, to do whatever it is to grab onto its prey. And then they'll use the stinging polyps to immobilize them. And here you have the reproductive polyps. So this is a colony of many, many different kinds of polyps. This example is Phasalia phasalis. All right, that's the Portuguese man-of-war. Let me unhide this. So you see that food capture, different types. This here is a pneumatophore. This like balloon-like structure. This actually helps it to move about when the wind blows it. So it acts as a sail. So this part is called a pneumatophore. Let us look at a little more of the Portuguese man-of-war structure. This is Gonianomus. You got to read that. So right now, oops. Oh, doggone it. I would have said worse words, but since I am being recorded, I wouldn't say these. So this here, which we are talking about, is our Portuguese man-of-war. And this is the pneumatophore, the one that's the bubble, which we're talking about. So here now is a more detailed structure in addition to your PowerPoint slide I just showed you. So these are the different types of polyps in this colony of the Portuguese man-of-war. All right, and they have different functions. They have different kinds of functions that they have to provide to make it hunt, support it, to feed, and to reproduce. All right. In class, we would have seen one of this in a container, like in chemicals that's sealed off. But this is a good representative schematic of our Portuguese man o' war of the Zalia species. All right, so let us go back to our PowerPoint because there's so much we have to do in this unit, but I want to give you a lot of details to help you understand the material better and to study better and do well in your exams. All right, so the next class is a Cyphozoa. These are the true jellyfish. All right, so what do they have is this amoeba-like cells that wander around under mesoglea. Again, this is diploblastic. And here in the Cyphozoa, the medusa stage is the dominant stage. They might have a polyp stage, but it's very tiny part of its life cycle, or none at all. So Cyphozoa is mostly, when you see them, is in the medusal stage. All right, so an example here is going to be on the Aurelia. So here, the medusal stage, this is life cycle. Let's start with where it is adult medusa. So what it does, once it's adult and sexually mature, it makes eggs and sperm because I believe they're dioecious. So you have your male and your female medusae. And then same thing again, like how we say in the Obelia, once the sperm fertilizes the egg and makes a planular larva, it's ciliated, and this is where it will find itself, implant itself in a substrate that is conducive for it to develop, and then it develop into a cyphostoma. So this kind of thing, you'll see it's like a, I don't know, like a champagne glass with things sticking out or whatever it is. And then from here, it will make it's called a strobila, where it's stacked up with ephyra. So as it makes it, it keeps pushing it up and up and up. So it makes it from this base, this part here in the bottom. And then it pushes it up where you have more and more, and it stacks up until it is the young medusa, which is called ephyra. And then it disperses and it will just go about its life as a mobile medusa. All right, so this is our Aurelia. Now, let us see. So they just make clones of itself. All right, so here we could see in the Aurelia that they're pretty. So you'll see the gonads in the gastric pouches. So this is when you look at it, and this is at angles, but this is a nice picture that when Caesar made these PowerPoints, he got it off the internet to show these. To get pictures like these is fantastic. So now let's go back here in our little notes to recap on our cyphozoans. And here is a jellyfish here. So this is like kind of dull. And of course, know this word staticus, which is, it allows it as a photo receptor and orientation sensor, so it knows whether it's up or down, So these sensory organs, allows it to go to places where it can, find food or go away from predators and all that stuff. So there's staticists. And what else we have? Later on, we'll talk about the Ropalia. So here is another schematic of the life cycle of Aurelia. So this is our sample, and this is a typical cyphozoan. So this is the same thing as the other slide. All right, so let's look at this here. This you need to know, very important. Medusal stage is the dominant stage, right? And the young medusa is called ephyra. So that word, you need to know that word too, ephyra. Okay, so here you could see this thing here. And of course, there's this special word, Ropalia or Ropalium, singular. This is where it's in this complex that's called, where you have the statocyst and all that stuff. And what else we have to do? And these are found in the periphery of the bell. And let's see what else I need to show you guys here. Because since we are not in class, I can't, we won't be able to see these things. Oh, it's not here. If we're in class, we will be able to see these things, look for them. Okay, so this is like looking down from the top or maybe beneath. So when you're looking down on it, like the aboral side of the medusa, so how many rhopalia do they have? So if they have one on each, the rhopalia, the statocyst, is in this complex that's called rhopalium, singular. So each one of these, the bell area, so 1 here, 2, 3, 4. Wait, let me put the dots. So, it's kind of like, oops, 2, 3. So I'm kind of doing the work for you guys so that you could see it. So these are the rhopalium, singular. Rhopalia total is 8, all right? So this is found here on the rim here. All right. So the radial canals, where the gonads found. So you see radial canals, they go toward the edge of the medusa. And here now, where the gonads are found in the gastric pouches. So you see them here. So they're the gonads, the 4 gonads, all right? And these, the oral arm, which are the tentacle. So you had seen that looking down. So if we're looking up, this is where we'll see the tentacle. So we are watching the mouth area. So we are watching it from the oral point of view. So you see the mouth. All right. So what else do I need to tell you guys here? Because I think this was in the final practical. But I do know that I'll give you guys these as your next, when you have to do your quiz, oral quiz. Okay, so the cyphostoma here replicates. That's the thing I was pointing out to you, by asexual budding. So they make clones of each other, and then it produces young medusa by sexual. So this here. This is where we talk about when they make the strobili here. Here is asexual reproduction, but this point here in the medusal stages is sexual reproduction, where it makes sperm and egg, and they will fertilize and make a new individual colony. All right, so let us go on to the next class, which are called the anthozoans. Anthozoans are the flowering animals. Antho means flower, so like anthophyta, like flower plant, or plants which are flowers. All right, so the anthozoans consist of corals and anemones, and the polyp stage is dominant. They can be colonial or solitary, and these are very important for coral reef buildings. They build the coral reefs, where like they have this symbiotic relationship with dinoflagellates, and the anemone also. All right, so not all dinoflagellates are bad, but we have a lot of very good ones. And what happens is, when now with the climate change and the oceans are warming up by a degree or 2, you get coral bleaching, which drives out the dinoflagellates and kill them. So then you'll see the corals are bleached, so they look white, and that's not good, because once the corals die, for them to to make protective coral reefs, they're very important as nursery for other kinds of organisms and animals. So this is very important that we are aware of our climate change, and this allows us to see how important it is to take care of the environment. and it affects everybody and everything. So the primary organisms here are the anthozoans that make the coral reefs. So they would leave coral structure, which is calcium carbonate. So that's what we see. Now in the anthozoans, we have these are the anemones. So this is, it looks like a flower animal. And this is a polyp stage. So it does look like this drawing here. The mouth is up here, which is called the oral end. And the part that's attached to the substrate is called the aboral end. All right. So this is an example of anthozoan, specifically anemone. All right. So now let's go back to this thing here I was showing you guys with this group. Let's talk about the hydrozoans. We talk about the cyphozoans, which is the Aurelia. And now we're talking about the anthozoans, and we're talking about anemones. Now let us talk about the next one, the corals in the anthozoans. So these are corals. You see how beautiful they look? And then what they leave behind is like the structure that kind of house the coral itself, the organism. So here now they're colorful because they're alive and all this stuff, but the dinoflagellates having a symbiotic relationship. And also, what else do I have to say for this here? And they're colonial. Let me see if I have that in the other stage here. Okay, so let us look at a schematic here of the anthozoans. So this is the anthozoans. It could be anemone or it could be the coral itself. So they have a pharynx. They have the tentacles around here, which sweeps in food into its mouth. So the tentacle will sweep in the food into its mouth. It goes down to the pharynx, and then you have this mesentery or mesentery, and this is really convoluted. Like it's kind of like cut up like where it has increase of surface area. So this tube brings down the food to this mesentery, and this is where the food will be absorbed. However, in the coral, since they exist in colonies, what happens is they have this structure where all these coral polyps exist on top of the limestone skeleton. So they leave that behind as they make their the skeleton calcium carbonate, and they have the special area here. Well, it's all throughout in the coral colony, and it's called a coenosorc. So the coenosorc connects all the polyps here, where all the food, when this one makes food and that one makes food, and each one of these coral polyp makes food, everybody feeds. So if one feeds, everybody feeds. And again, mesenterial gut, they have septa, which helps increase the surface area of food absorption. And then, of course, the mouth is facing up, and you have the tentacles. So this is a cross section of our coral polyp. All right. What else we have to do? And then sometimes, you know, like when you see they all open up like here, it's in all its glory. But again, if they're disturbed or maybe they're not feeding, they withdraw these tentacles and it will just close up. All right. So next slide is 4.3. So we have to go real fast. The last class in our cnidarians is cubozoa or cubozoans. They consist of the sea wasps and the box jellyfish. So the jellyfish look like a box. They're not really jellyfish, but they call them box jellyfish. They look like a box. The cyphozoans are the true jellyfish. All right. So here we have tentacles again arising from the four corners of the body. So it's a box-like architecture it has. And then they have these highly developed sensory organs again, which are called the rhopalia. And then here we can see these sensory organs. See this rhopalia here. And within it, you have the pit eye and the slit eye. within this rhopalium, and you have upper lens and lower lenses. So this here now, this here that has been, like different from all the other eye evolution in animals, like how they can sense they evolve independently. So here you could see the rhopalium here, and all that stuff. So let us look at the cubozoa, the box jellyfish. Well, actually, that's it, which we need to talk about. So let us look at the slide, the PDF here to just recap. So we did talk about the coral and the anemone, which is in the anthozoans. So this, okay, our example is Metridium. Know this one here. This is the thing we would have observed in class when we used to do our final practical. The anemone, this is an anemone. It is an anthozoan. Please make note of that. And this is a polyp. So this here would show the different the partitions, mesenteries, and the ovaries where the gonads found in the partition. All right. So what else I need to tell you this about? And again, the theme here again, nematocyst. That makes it, a cnidarian. And this is our example of our anthozoans. And this Metridium is an anemone. All right. What else? I believe there might be a question like that in a final practical. So please, make a notation now. So when you're studying. And corals, we talked about this already, right? Of the different Sinosarc and all that stuff. And I said if they're not feeding or they're disturbed, they'll retract the polyp, So here it's expanded. And the Sinosarc, which we talk about, one feed and everybody feeds. So what else? Feeding. And you have the septa, which increases the surface area. Corallite is the leftover skeletal structure where the corals would have been residing in. All right, cubozoas, we talk about the sea wasp and jellyfish. And our example is Caronex flexii. But since we are not doing the class, I don't think you guys are expected to know that. And this is another schematic here, the spermatophore and all that stuff. So read up on this. And of course, it said the box jellyfish, they're very deadly. Like, for example, in what do you call it? In Australia, there are a lot of deaths that resulted from there. All right, so next we are going to talk about ctenophores. These are the comb jellyfish. Do they call them jellyfish? Let me see what they call them. Ctenophores. Well, they have the comb, the teens. That makes them called ctenophores. So where's the word we're looking for? Okay, here, teens. These are the words, how you could study that how they derive their names from. All right? So ctenophore refer to the combs, which are called the teens, and they fuse the group of fused cilia, and they form this kind of plate-like arrangements in 8 rows. All right. So here, let us look at it quickly. So this year, they said ctenophores may be triploblastic because they have true muscles. So you got to read up on this, and then they have a past digestive system is not. You remember we were talking about the others, which the hydra, which is, and the cnidarians, which is a one-hole gut sac. They have a pass-through gut. So they have a mouth at one end. You need to know that important, and an anal pore opening on the other. So here you see the mouth, and here you see the anal pore. All right, so it's a pass-through gut. And here is the oral direction, and here's the aboral. All right, so these are the features that you'll see here, and the comb rows, which are 8 of them. All right, so what else I need to say? So just read up on that. Okay, so now we are moving on to protostomes. What we just covered, and I know you guys are probably like, like really like dazed now with all this information. So now we just covered that part of our diploblastic animals. Now we are moving on to triploblastic animals. So there are two lineages of triploblastic animals, protostomes and deuterostomes. So today we will cover in our this part of the triploblastic animals. Here we'll do this part here. We just did the diploblastic, so we did that. We'll talk about flatworms, which is part of the Lophotrochozoans. All right. So the Lophotrochozoans here are this group here. So this figure you need to know. So this is the Lophotrochozoan lineage in the protostomes, and it consists of arrowworms, bryozoans, flatworms, rotifers, ribbonworms, brachiopods, phoronids, annelids, and mollusks. So since we are doing some of the protostomes, not all of them in the Lophotrochozoans, we will, because next week we'll do the Lophotrochozoans, and the week after we'll do the ecdysozoans, and then deuterostomes 1 and deuterostomes 2. So you have 4 other big sections to do, which are in units 20. Well, 20 would be Lophotrochozoans. 21 is going to be ecdysozoans. 22 is going to be deuterostomes 1, and 23, which is your last unit, will be on deuterostomes 2. All right. So this here, what we're going to do is talk about the flatworms today in our Lophotrochozoans. So that's why we said some protostomes today. All right. So now, what is a flatworm? Here it is flat. All right. So here, this is what we're going to do today in this unit 19. Since I gave you guys this PDF kind of late, not enough time for you guys to read through it, but I would trust you guys will read this at least 3 times to get some knowledge into your head, and you have already covered this in your lecture. All right. So what again are they? What kind of coelom do they have? They do not have a coelom, so they are acoelomate. All right. So I just saw the word here, flat. What else? And then they do not have a respiratory system because they're flat. The gas exchanges, it could diffuse out of its body because it's so flat. And then in the a lot of them are parasitic, but some of them are free living. So their lifestyle here, they could be free living, but if they're free living, they're mostly marine and freshwater. And some of them could be terrestrial, but the majority are parasitic, all right, which includes the flukes and the tapeworms. So we'll talk about those 2 clades in this class. So we have 18 minutes, so I have to go through it really quickly. All right, and they say they have some kind of form of cephalization, which means there's a head formation. So which means the head is forward and they have a bilateral symmetry. So that allows it bilateral symmetrical organisms. All right, so these are the key words here, but I'm looking for the word acoelom, and I saw it, and then it's like, where did it go? Let me quickly scan through this. Flat, no respiratory system at the same time. Just understand they're acoelomate. So when you're looking through this, I thought I saw it in these couple of paragraphs, and it kills me. It's like seeing them, scanning it quickly, and now I can't see it. Can't find it. There we go. Okay, so we were talking about the different types: acoelomate, coelomate, or coelomate. Acoelomate, pseudocoelomate, or coelomate. That's when we're talking about all of them. But in this particular case of the flatworms, they are acoelomate. They do not have a coelom. All right. So let me see. Might be in the other group. No. I hate it when I can't find it. I saw it when I did a quick scan, you know, it's like, and then it's like, and it's killing me. It's like, I got to show you guys this so that you could see it in the recording. Let me see this. Oh, here we go. So they're acoelomate, all right? But they are in the lophotrochozoans. Then we'll see why they have this thing here, all right? So these are the things. The reason is because they have under here bilateral symmetry, 3 embryonic layers. So these are triploblastic, ectoderm, mesoderm, and endoderm. And then they have cilial locomotion at one stage of its life, either when it's like going through the larval stage, when they're developing, adult stage, or at one point they have cilia to help them move, or they have cilia around it for locomotion. All right. So now this here is the 4 clades or groups of flatworms. Let me make it bigger for you guys to see. Okay, it's very important that you know them and it's split such a way that you can memorize them. We will talk about all of these in this class. And if I don't finish, I'll continue recording, but you guys certainly do not need to stick around, you know, because I know this class finishes at 4:30, but I will not, you know, stop there. I'll just continue recording, all right? So we have turbellaria. This is important. They're called the planarians, or they're just called flatworm. You know, they're flat, but it's a flatworm. So the best way to call them is turbellaria, you know, by its group, or planarians. Now, what are the 4? These are mostly found free living, you know, and then they have ventral epidermis and they have mucus in the bottom of its, you know, like to help it, and the ventral part in the epidermis. The mucus allows them to glide easier for locomotion and cilia. So they do have some freshwater species. In class, we would have done the experiment with a planaria where we cut the head down the middle to 1/5 of its body, and we want to see it regenerates 2 heads. But it never happened because I think if you don't do a proper dissection, it will not develop into 2 heads because it shows that it can regenerate body parts. Okay, so now the life cycle, hermaphroditic individuals, but they do not self-fertilize, they cross-fertilize, all right? Our example is Dugesia as our flatworm, which we would have done in class. The next clade is Trematoda, which are called the flukes. We'll talk about that. Our example is Clonorchis sinensis and Schistosoma. That's the blood fluke. We have the liver flukes and the blood flukes. Now, when we say flukes, we are not talking about a fish. You know, flukes, as they call them flukes. These are flukes as in not good parasitic flatworms. All right. There is another, this group, Monogenia, but we won't talk about too much in this class. But, you know, like I say, that means it has 1 host. All right. So this one, not so much. But we'll talk about the Cestoda, which consists of the tapeworms. So tapeworms are real flat. But when you see the flukes, some of them look flat, but not totally flat. So now, in our example of our species for tapeworm Cestoda, it's Tinea. All right, so these are the stuff that we need to kind of focus on. Oops, sorry. Kind of like my hand is not doing it properly. All right, so this and schistosoma. This is very important because you can either know that it is hermaphroditic or dioecious. All right, so let us go into our slides here because there's quite a bit to go through. All right, we talk about ctenophores. We went through that. Hold on, ctenophores. Okay, so this you'll have. We talk about it in the other slide. And these are what it looks like. So now let us go on to our flatworms in our triple blastic animals, which is in the Lophotrochozoan of flatworms. All right. So it's called, the phylum is called Platyhelminthes. You know this here. It's important. This is a phylum. Platy is flat, plano, helminth, worm. So Platyhelminthes, Lophotrochozoan, triple blastic, acoelomate. All right. So you need to write all these things down. The three classes that we'll cover, all groups, as I say, flatworms, but actually it is Turbellaria. Write it down. Turbellaria. You know, it's like trying to remember. All right, so don't confuse it. I call it flatworms, but it's actually Turbellaria. So it's flatworm or flatworm planaria. Let me write it here. Planarian flatworms, and that's Turbellaria. Wait, let me reverse it. Sorry, guys. I want to reverse it so that we are consistent. Undo. Hell, I messed up. I'm supposed to do Control C. Then take this out. So I'll put it here. Okay, so then it is consistent to our three groups, all right? Goodness, it's giving me problems. Backspace. Okay, so this kind of make you understand that these are the planarian flatworms, are not flatworms. All right, so let us go through this. So the flatworms, which is the, again, Turbellaria flatworms. Let me, so I can make it specific. Otherwise, you guys will kind of like get confused. Let's make it smaller. You don't need it so big. Oh. Like trying to sort these things out at a smaller, whoa, to make it like bigger. All right, so how do I make it, bring it up? Okay, here we go. All right, so here now is our first group, Turbellarians. All right, so here, this is what they look like. They're free living. They're acoelomates. As I say, I spent all this time, they're flat. They do not have segments, because we'll see that in the earthworm where they're segmented. They do not have any respiratory system and that sort because they're so flat. So they have a special expiratory system that's called a protonephridial system. So that get rid of its nitrogenous waste and all that stuff. We'll see that in the following slide. And the thing is, as we say, they're tropoplastic. Most of them are parasites. But this one here, the planaria dugesia, is free living. So this one is free living. This example here, here you see it's a triangular head and the pair of eyes or small tentacles that you'll see here. So this is our example. Dugesia is our example. Let me see if I could add this on in class, because since we are not doing our class work, Dugesia species. All right. So here you have to add more stuff like what I didn't have before when I did it in class. Okay. So now let us look at the schematic of the flatworm. This is these are taken off the internet, and you know, like the stain them and all. So this is the head. This is the anterior part. So this is anterior. You have the eyes, the ocellus, the eye spots. Look at where it obtains its food. The pharynx is all like halfway down its body. It's very muscular in this area. So if you do a cross section here from this area, like if you do a cross section from here through the pharynx, you'll see this muscular part here. This is the one that you'll find all the way over here when you do your dissection, all right? So this is, I want you guys to see that. So if you cut it through here, this is what you'll see here. If you cut it more in the front here, this is this part, all right? And if you cut it more to the back end of it, you will see the different parts of this flatworm doogesia. So what happens here now, you see this center here is the intestinal part that it goes down. It does not have a zone, all right, because it's acoelomate. This is a pharyngeal area. So this and this is the same, all right? So let's see. So this here is a cross section here. This is a cross section. And this is where it comes through the pharyngeal. So next thing you see is like they have this special protonephridial system. So let me see if I have it here. No, I don't. So in our protonephridial system, let's go back into this page here. See, this is what it looks like. And here is, this is very important that you guys look at this slide really well, excretory system. So this is the protonephridial system where it gets rid of all this nitrogenous waste. So you see all these things that looks like, you know, like network of flame cells. So you need to know the flame cells. So how it gets rid of its nitrogenous waste, it has an excretory pore, all right? And here, this is where you get a bigger, for the, here you see the flame cells, you get a more exaggerated view. So this is the excretory system. Now the sensory system, you have the eyes, the eye cup and all that stuff, and a brain area. And now the digestive system, as I say, it's like a blind gut. You have the pharyngeal area, which, you know, it has an opening. And then the food, all the dark areas that looks like, you know, like making an ink blot. This is the digestive system. So you see the intestinal area here, where it's pointing to. And then again, if we look at it in the slide here, you can see the shape here. And what else I need to show you guys, where we said it has, the system is the protonephridial system with the flame cells. And then you'll see the digestive system. All right, what else I need to show you guys here? And then this is a cross section from this area here that you'll see it has longitudinal muscles and circular muscles. And that was, it arrived from the mesoderm cells, all right? You see the nerve cord. So this, you know, transmit, you have the dorsal ventral muscles, mesenchyme. So it does not, since it's acoelomate, here it has parenchyma. And what else I need to show you guys? Circular muscles. All right, so this is how it moves around. It undulates. All right, so let us move on to the next slide because we have a lot to cover. Okay, that was something you look at. The next group is the trematodes. The trematodes are the flukes. There are two groups of trematodes. They're called digenia, which means it has two or more hosts for its life cycle. And these are found like intestinal parasites. And you have monogenia, which are the one host in its life cycle for the flukes. And those are ectoparasites, which we'll see in the gills of fish. So let us look at this group here of our fluke. So this is the schematic of our fluke here that it would show. This here, I believe it's a liver fluke. So the liver fluke here, you see it has an oral sucker, so it gets its food from its host. It attaches to the host and then it'll suck, the sucker. So they will suck the food, they'll swim, wherever they are located. Like they have the liver fluke, which will reside in the liver, but of course they will be passed out in the feces of its host. And then you have the blood flukes. So let us look at this schematic of this fluke here. So this is a life cycle where it has two or more hosts. So you have the human host and you have snails here. So what happens is if food is not properly prepared, like let's say for instance fish. So here now we have snails. So let us look at this here. Snails. Let's start from this area here. So this is ingested or it takes into the human, and the egg cyst will go into the duodenum, and then it penetrates into the intestinal walls. It goes into abdominal cavity. It penetrates into the liver. So this in the liver fluke will develop. So what it does while this it's in the human host, of course, it's getting all its food and all its stuff, and it will make eggs and it will be passed out in the feces of the human. And now if these feces got in an area when the embryonated eggs become embryonated, they will develop into merasidium in the snail, because the snail will crawl around wherever, let's say for instance, that somebody take a crap, I have to say it in that way, or defecate in, not in a regular bathroom, but out in the field, and there are snails around and water plants. It will pick up these eggs and they will develop in the snail into merasidium in the snail, and it will further develop into sporcis, redia, and cercaria, where the snail will, as I say, eject this into the water, and when a fish feeds on this, it infects it. And when you eat the meat of the fish that's not well prepared, that to kill the cercaria, it will infect the human again. So this is the life cycle of Trematoda. So let us look at the life cycle here. As we said, the same thing. So our example of this is the Chinese liver fluke. And the slide we would have looked at in class was Opisthochus, and the previous name is Clinorchus. So it's like either, they're interchangeable. So you see it's multiple hosts that it needs. So this is Digenia in the fluke group. So this is the same thing I talked about in the former slide, the previous slide. So this here is the liver fluke, and these are our example that we would have seen in class. Now we have a different fluke here now, schistosoma. But let me come back here to see, I believe they are hermaphroditic, the previous one, but this one here is going to be, I'm coming back here on it, all right? So bear with me here. Okay, so coming back onto this, let's go back and if you look onto page 347, let's go back into 347 because I want you to see this thing and understand it without going back and forth and looking to find where you got to find these things. Okay, so this is what plain thing here. So now this is hermaphroditic, all right? This liver fluke is hermaphroditic. Okay, wait, somebody just buzzed. Okay, Z, as I was saying that I will continue recording. If you guys need to go, please go ahead, and then you guys could see the recording further, all right? Because I just don't want to stop here and the next week you don't have your, how to call it, the whole lesson. As I was saying, I will flow into the very end because I think there's like 15 more minutes But for those of you guys who want to stay, you're welcome to do that. But by all means, I do not need you guys to stick around, okay? And then, we'll flow into our office hour, all right? So when we finish, we'll just close off there and go into the office hour part, okay? So let us continue in the work. I know it's a lot to cover. All right, you're welcome, Grace and Edina. And for those of you guys who still have your attention. So this here is, I'm going back to slide number 39. This is the liver fluke. They are hermaphroditic. They do have both male and female sexual organs for Clonorchis, and I failed to tell you guys that. So you see testes and ovaries. So they're hermaphroditic, all right? That's what I need to point out to you guys. So they kind of like fertilize their eggs, and then once they disperse it through the feces. All right, so coming back into our slide, somebody said something. Wait, hold on, let me come back. Okay, I guess some people left because they have to go for classes, and I don't blame you guys, but it flows over because I need to show you guys more things. But the schistosoma, this is the blood fluke, all right? The other prior one is the liver fluke. Now, this is the blood fluke. This is the life cycle of it. This is dioecious. They're separately sexed. They have a male schistosoma, and you have a female schistosoma, all right? So now, this is a life cycle. It needs more than one host. So same thing again, like how it gets into the human. If you run around barefooted or whatever it is, in the urine, it's like you step onto it, and the heat. So you follow this life cycle of the schistosoma. All right. So in the schistosoma, this is what it looks like. So in class, this is what you would see. This one here is a female. The female is embedded in the groove of the male. The male is the bigger one on the outside. So this one here, let me do the slideshow thingy. So this here, wait, hold on, pointer. Come back on the slide. This here is a male. This on the outside is a male. All right. The female is this small one in the groove of the male. So the female is small, and they're paired for life because it's hard to find a mate, especially when you're in your hosts and flowing around in the blood. So here again, this here, the smaller one, this is the female, and this is in the groove of the male. The male is on the outside, the female is on the inside. Same thing here. All right. So now let us go back into this full screen. So here we go to the next slide. This is the monogean fluke. So this is a parasitic fluke on the gills of a fish. All right. So here we have just one host. And let's go on to the next slide. Okay, that was our prematoda. Now we are moving to cestoda. Cestoda are the tapeworms. All right. So tapeworms, they do have distinct segmentation. They lost most of their organs because they want to be highly reproductive. And they have a protective epidermis because they live in the intestines of the hosts. So in tapeworms, like Elatina is our example, when they are in our intestines, if we're infected, the acid could eat away on the epidermis. So they have a protective epidermis exterior. All right, so let us continue with tapeworms. Now, the head, the scolex of the tapeworms, the scolex of the tapeworm has hooks on them. So that attaches to the lining of the gut of the host, like for example, the human, or they have them in the cows. They have tapeworms too. And then what happens is they have these sections that's called proglottids because they're segmented that grow behind the head. So once they grow behind the head, they produce more sections, and the older the section, the further down it will be away from the head, and they break off in the gut of the hosts. So when the hosts defecate, that piece comes out, and they have the fertilized eggs, which can develop, and then again they will find another host to develop and spread, live onward. So here now, in the proglottid, it's male first, so sequential hermaphroditic. It will be male first, and as they age, it will become female. They can make female gametes. And I said the proglottid will separate from the rest of the body, the very, very end, and they have these eggs which are fertilized already, and it will be expelled through the through its feces of the host feces, and then it infects others. So we'll see the life cycle of this tapeworm in a little while. So this is what the tapeworm will look like in our how to call it. If you were to look at the slide, this is the head, which is called the scolex. They have the hooks that hook onto the gut, the lining of the gut of the host, and these are proglottids. So these are sections which could break off on the very, very end. So this is showing that it has both male and female sexual reproductive organs, the testes and the ovaries. The testes are first formed, and then later on they will develop ovaries. All right, so they become sequential hermaphrodites. And what else I need to say? So we have one more slide. So let us look at the life cycle of this. So here we have, let's say for instance, our tapeworm here. Let's look at the life cycle. So we have Tinea saginata and Tinea solium. Yeah, I can see it. So here when the proglottids are gravid, that means the eggs are in this proglottid part of this tapeworm. See how it is? And tapeworms are cestoda. This piece breaks off, the very, very end. This is a piece in the end. The head is all the way up here. This is the scolex, okay? The head is all the way up here. And this here is the piece that breaks off. This piece that breaks off when the human, pass the feces with this. So for instance, let's say somebody take a crap out in the field in a pasture, and this is, it's like on the grass, and the pigs or the cows that eat the grass eat these grass with the feces that's infected with these gravid proglottids. It will, these onconospheres would hatch in the pig or the cows, depending on the species of Tinea. And then what it does is will go through, it will penetrate the intestine wall and it'll circulate to the muscles, which is in the meat of the pig or the cow. And if you do not cook this properly, like enough heat to kill these oncospheres, they'll develop in the cystical kind of cyst in the muscles. And once you do develop in the muscles and you don't cook that meat properly, and the humans eat this meat that has this infected, cystoceri, this will again go through a cycle and it will affect the host, which is the human, where the tapeworm will develop in the intestine of the human. All right. So this terminates our, I took 13 minutes over, so it's not so bad, of our unit 19 of our lower invertebrates and some protostomes. So we just finished covering our flatworms, so which consists of our 3 groups that we talk about, the turbellarians, the trematodas, and the cestodas. So let me type in here our example because I'll give you guys this. So example here, we have tinea. So I will probably increase it more. Tinea, let's spell it right, species. That's what we would have done in class of our thing. So class, this is the end of this lecture or recitation or punishment. Let's put it this way. And hopefully you guys go through it. It's long, it's tedious and all that stuff, but by all means, you can stop, pause, or whatever it is, do some more reading. All right, so class, for those of you guys who want to see me, yes, Sarah, I'm going to send out a PowerPoint to all you guys, okay? So that you guys could study off of it. I already give you guys all the other PowerPoints so that you guys have to study for your final practical. All right, so what else I need to tell you guys? All right, so I'm gonna stop the recording here and then we'll start our office hour after I have a little drink. Oh, PowerPoints, I will put the PowerPoint in the weeks folder. How is that? Okay, thanks, Suleiman. Have a great day, rest of the day, all right? So everybody, have a wonderful weekend and study up, keep studying, do well, stay safe, all right? Okay, guys, I'll stop recording, okay? Let me see, okay, let me put that, and have a great weekend. And study up. Oh, I forget to put a G. All right, guys, I'm stopping the recording. And then for those of you guys who want to see me, come on